The overall goal of this procedure is the reliable investigation of the adsorption capacity of selective iron hydroxide containing materials, such as granular ferric hydroxide, regarding phosphonates with a low workload and significant cost savings compared to conventional methods.
This method can help answer key questions on how well phosphonates can be technically eliminated from industrial wastewater, such as membrane concentrates, in which phosphonates are present as antiscalants.
The main advantage of this modified phosphorus determination using 10 milliliter vials and a thermostat is that more results can be produced within shorter time compared to the conventional method.
Demonstrating the procedure will be Raith-Bausch, a technician from our laboratory.
To begin the experiment, select one or more buffers with suitable properties for this technique that buffer the pH range or ranges of interest. For each buffer to be prepared, weigh or pipette the needed quantity of solute to prepare a 0.01 mole per liter solution into a volumetric flask. Add reverse osmosis treated deionized water until the flask is about 3/4 full. Add the needed volume of a phosphonate stock solution with a concentration of 1 gram of phosphorus per liter to achieve the target phosphorus concentration in the buffer solution. Then fill the flask to the ring mark with water. Stir the mixture until the solute has completely dissolved, or for 5 minutes for a liquid solute. Then, transfer the buffer solution to a labeled glass bottle. Adjust the pH of the solution with either hydrochloric acid or sodium hydroxide to the desired pH value.